Good afternoon. Good afternoon. How you guys doing? So far, so good? Good. You have the exam this Thursday, right? Excellent. I know the antiviral today is not covered in the exam, correct? It's going to be only the antifungal. So do you have any questions from the previous lecture then? Have you studied yet? It's probably too early. Do you have any questions from last time or anything in general? So far, so good? Is the quiz, is it the exam easy or the quiz? When is the quiz? Today? I don't know. You're gonna let me know. It should be easy. It's like, isn't it like open book, right? It's, you take it at home, right? And you have like 30 minutes? Okay. So you could cheat. No. Is it open book?
Yeah, take homes.
Yeah, so it's not cheating. It's good. So, but I just have to be, I guess, prepared and aware of the materials before you can look at them. All right, I'm getting in trouble. Okay, so today we'll talk about antivirals. So actually, we should be talking about the antiretrovirals. But you know, we cannot talk about that without introducing the entire, you know, antivirals. So we'll have to talk about that, and then we'll specifically talk about the antiretrovirals. We'll also talk about the when we get to hepatitis, we'll also talk about some of the antivirals for hepatitis. Okay, but for now, we'll just give a brief introduction in general about the different kinds of antivirals, and then specifically, I mean, look at the drugs that are used for HIV. Have you learned antivirals at all? This is the first time, right? The virus life cycle and antivirals.
We only know one thing, acyclovir.
Acyclovir. So, like, have you studied any other courses the life cycle of the viruses and the antivirals? No. Okay, so this should be fun then. Okay, so feel free to stop me for explanation, okay? All right, so to start with, viruses, just a brief introduction to viruses, how they are different from bacteria. So unlike bacteria, they're actually viruses are dependent on the host. They cannot survive alone. They have to, I guess, penetrate the host cell and then use the host cell to produce other viruses, okay? So they are obligate intracellular parasites. So they are parasites. They cannot live on their own. Okay. They are defined as submicroscopic entities that consist of a single nucleic acid. So the nucleic acid could be RNA or DNA. Okay, and so we have RNA viruses and we have DNA viruses, and they are surrounded by protein coat. Okay. So this is a coat that has all the proteins that are needed for the life cycle of the virus, as we will see, okay? Because the virus is gonna need to insert this nucleic acid into the host DNA, and then once it's replicated, once it gets its proteins, it has to cut them and so forth. So there is some processes that the virus need to perform. And so the virus is nothing but a piece of nucleic acid, which could be DNA, RNA, plus proteins that are needed to serve its purpose. Make sense? Okay. And then, of course, it's capable of replicating in the living cells. You know, it will need a living cell to be able to replicate. And that's how it damages the living cell. By replicating so much, it occupies the function of this cell, and then this living cell would lose its function and die. They are actually much more smaller than prokaryotes such as bacteria. They range in size from 20 to 300 nanometer. I think I have a picture just tells you relatively how, you know, the size difference. So this shows E. coli bacteria, and these show the sizes of the viruses, just so you get an idea of how small the viruses are. So the infective part, I mean the virus, when they are extracellular, we call them when they're outside the cell, we call them virion. Okay, the infective extracellular virus is called virion, but once they're inside the cell, we call them viruses. We have viruses that attacks human. We have viruses that attacks bacteria. We also have viruses that attacks fungi. Those that attack the bacteria, they're called bacteriophage. Okay, those that attack the fungi are called mycophage. Okay, we even have viruses that attack other viruses, and they're called virophages. Okay, have you guys heard of? You guys might remember CRISPR technology. You guys heard of CRISPR technology? What does CRISPR technology do? CRISPR technology, what does it do? You've heard of CRISPR technique? What's the advantage of that technique? What does it do? What's the main, I guess, job of CRISPR technique? Why is it such a famous technique and it's like breakthrough? They received the Nobel Prize, I think, a year or 2 years ago, people who work on CRISPR. So CRISPR is a gene editing tool. It could actually cut the genes that are causing a disease. It could also substitute a gene with another. So there are different ways CRISPR can work. One of them is cutting the gene that's responsible for the disease. That's very helpful, right? I mean, after all, that's where the diseases are coming from. So if you cut those genes, you eliminate the disease, okay? Why did I mention that? Because the whole idea of CRISPR came from bacteriophages. Those are bacteria that, so there are viruses that attacks bacteria, and so the bacteria has a mechanism of defense. Once the virus attacks it, it takes a copy of the virus and saves it. And so when the virus comes again, it recognizes it and attacks it. This machine was used for the purpose of CRISPR technique. How is that? They have used the proteins that uses the virus DNA and cut them and insert them into its own DNA to mimic this technique such that it could be used to edit human genes, human DNA, okay? So in short, the bacteria takes a copy of the virus DNA or RNA and then saves it someplace where it has to use to identify the future attacks. And so this machinery in the bacteria has been used to design CRISPR technique. It's very nice, right? Okay, I hope you agree with me. Any questions? All right, so the origin of the viruses is unclear. It's believed to evolve from plasmids, while others might have evolved from bacteria. There are like about 5,000 species of viruses have been discovered. Okay, let's look at the structure of the virus. We have three main components. We have something called the capsid, okay? We have the envelope, and then we have the nucleic acid. So this is the capsid, which is the protein that has, it's like a bunch of proteins that are surrounding the nucleic acid, okay? So this is the nucleic acid. And then some of the viruses have envelope. So we have three components. Now some other viruses might have the capsid, the DNA, and they might have a vehicle that is responsible for inserting the content, its DNA content, into the cell. Okay, so the capsid is a protein shell that encloses the nucleic acid. With its enclosed nucleic acid is when you have the nucleic acid enclosed, it's called the nucleocapsid. So we have capsid. When you add the nucleic acid, it forms nucleocapsid. Okay, this shell is composed of protein organized in subunits called capsomers. So these are the proteins that the virus will need to attack the DNA of the host and perform all these functions for the life cycle, as we'll see soon. We also have an envelope. This envelope is similar to our membrane, okay? It's like a phospholipid bilayer, okay? It also has some glucose, so it's a glycoprotein. These are essential for communicating with the host cell and initiating the viral fusion, as we will see in the life cycle, okay? And then, of course, the nucleic acid, we have DNA, nucleic acid, and we have RNA. So you may have RNA-based viruses, and you may have DNA viruses. Okay, so far so good. Any questions? Yes.
Going back to like what you mentioned about CRISPR, right? Like with the bacteria, I find that interesting. Is this like how is there like how we implement like medications basically with the same way how bacteriophages think in that way? Like, okay, are there medications that we're trying to build up that has that same exact mechanisms with bacteriophages?
CRISPR medications or medications in general?
Like just like the CRISPR medications, are we like going steps further to going like making medications that do that same mechanism?
That's what CRISPR technique is doing. So CRISPR is nothing but a bunch of proteins that with a DNA. And so these proteins, the idea of having these proteins were adapted from the bacteriophage. So these proteins are inserted into the human body, and they go cell by cell, and then they cut the DNA, either just cut it and leave it, but that's where the piece of that's generating the disease, or they might cut the DNA and insert another piece of DNA, okay? And so they could substitute the DNA, even though that's more challenging. It's not been like in practice approved yet. It's still in research. The idea of inserting the DNA is still in research. But what's been applied and it's been successful in treating diseases is actually cutting the DNA and removing the piece of the DNA that's causing diseases. And they've been so successful in treating certain diseases. Okay.
That's interesting, right? Do you think that this would kind of go up to the step of human genomics in some way, like pharmacogenomics?
So pharmacogenomics is something else. It actually has to do with how different people would have, because they have different DNAs, so they will have different enzymes, different metabolism, different response to the drugs, and so we have inter-individual variations, okay? So before you could choose the drug that best suits a patient, you may check their DNA, make sure that they have the enzymes needed or they have the receptors that could act, mostly enzymes,
Okay.
okay?
All right, thanks.
Sure. So far, so good? All right, let's talk about types of viruses. We briefly talked about it. There's a DNA virus, which has either a single or most of the time double-strand DNA. Examples are the pox viruses, herpes, and adenovirus papilloma. And we have some RNA viruses, largely are single-stranded RNA. Okay, so those RNAs cannot be inserted into the DNA. So when the virus has an RNA, it has to be converted into the DNA, and then that piece of DNA will be inserted into the host DNA. You see the difference, guys? So there is an additional step needed in the case of the RNA viruses. Why is that? Yes, Eric?
I was thinking like maybe it's because like the DNA then could get incorporated into like the cells and the nucleus.
So the DNA virus is ready. You just has to be inserted into the host DNA. And then the host will produce all the proteins that are needed for the virus life cycle and replication. But if you have an RNA, then it has to be converted into the DNA first, and then that DNA would be inserted into the host DNA. Make sense? So there's an extra step there, additional step, okay? We'll talk about the enzymes soon, but it's called reverse transcriptase. Remember the transcription translation process in the protein synthesis? We have DNA going to RNA. using transcriptase enzyme. So there is something called reverse transcriptase that could convert the RNA back to the DNA, and then it will be inserted. Okay. All right. And these are the different examples on the RNA DNA viruses. Honestly, for my portion of the exam, I don't expect you to memorize which one is which, but I think you might want to know that for the pharmacotherapy. And we talked about the relative size, of course. Okay, now so far so good. Any questions? Excellent. All right, let's talk about the life cycle of the virus. So first of all, we have an attachment. The virus attaches itself to the surface of the cell. Okay, and then we have entry, where the virus inserts its genetic material, or it could enter itself, like the entire virus would enter inside the cell. And then we have the genome replication and expression. So that's when the RNA will be converted into the DNA. The DNA will be inserted into the DNA of the host, and then the host will be producing proteins on behalf of the virus. And those proteins will be used for producing more and more viruses to move and go and attack the next cell. So throughout this process, once these proteins are synthesized, there's something called assembly. So when the host synthesizes all these proteins on behalf of the virus, all these proteins will have to assemble along with other copies of the RNA and DNA for the virus, and they would assemble into a new virus. So we'll have new viral particles. So one viral enters and process all these, and they will end up having multiple viruses. And then, of course, the last step is the release of these viruses to go and infect other cells. And by occupying the cell is doing nothing except producing those viruses and those proteins, and so it's not doing its function. Okay? Everything is good so far about the life cycle? And so just a brief hint. As you could imagine, antivirals will be acting on these steps, preventing the fusion, preventing the uncoating, preventing the multiplication, and preventing protein synthesis, preventing the release. Okay, we'll get into, this is just very general, we'll get into more details. Okay, so now as far as the viral entry, we have two kinds of viruses. Some of them have envelope, others don't. And so those that have envelope, there are two ways that they could enter the cell. Those that do not have envelope also have two ways to enter the cell. The first one is something called, is something similar to endocytosis, where the membrane of the virus fuses itself to the membrane of the host, and then the content of the virus gets inside the cell. The second type is like forming like a vesicle around the virus, and then this vesicle will break, and then the virus will be released. Okay. The same thing here, or in the case of the non-enveloped viruses, the virus could form this envelope, and then there is some proteins that attach to the surface of this envelope that eventually leads to the release of the virus. Or you might have like an endocytosis where a vesicle is formed, and then the vesicle lies, and then the virus would be released. Okay. Questions? Yes, Eric. I'm sorry.
When you were talking about like the last one, right? Like I see like nuclear envelope, and then like I see like the virus kind of releasing its contents at the end over there. Like, what's that thing that is able to go and allow that to go and release like the DNA?
Oh, so in the host cell, remember we have the cell and then we have nucleus. And so the nucleus has a nuclear envelope. So the virus could be inserted into the nuclear envelope because that's where the DNA of the host is located. So we want the virus nucleic acid, the virus genetic materials to go inside the nucleus. Okay? Make sense?
Yep, thank you.
Sure. Yes, you have a question, sorry. Third mechanism involves, the only difference with the third mechanism is you actually have some proteins here in green that facilitate the engulfing or the inserting the virus inside the cell, and then another protein here causes or actually the content of the virus could be released directly from the virus into the inside of the cell. The first one is different. The first one, you have this membrane is fusing into this membrane. So you would see part of the membrane of the host now has the membrane of the virus. Okay. You're welcome. So far, so good. Okay. All right. So we talked about virus entry. I think there is another slide about virus exit. But before that, just to keep in mind, these processes, as you can tell, have proteins on the virus as well as proteins on the surface of the cell that communicate for this entry to happen. And so this is a list of these proteins. Why is this important? Because those are kind of receptors that will be targeting to prevent the entry of the virus. Okay, so you don't have to memorize anything in this table. This is just for your own information. So in the case of the HIV, we have those host cell receptors, okay, that are present at the surface of the membrane. And so as you can imagine, we could interfere with the interaction between the virus and those receptors. So the virus have to interact with the cell host. So we could actually design drugs that block this interaction. Okay, I highlighted two of them because we'll use them in the later in today's class. So we have CD4 glycoprotein, we have CCR5. Just keep these in mind. We'll get to them later when we talk about drugs that prevents the infusion, of the fusion, of the virus. Okay. So far, so good. Okay. All right, now let's talk about the exit. So when the virus is assembled and ready to leave with its protein and its viral content, remember we need capsid protein and we need the viral chromosome or nucleic acids, which could be DNA or RNA. It assembles and now it's ready to leave. So there is something called budding, where the virus would get to the membrane and then this, vessel forms and leaves the cell and attacks the next one. Okay. Now notice that there is some proteins or receptors in green here at the surface. So these actually help with the formation of this vesicle, and when it leaves, they actually help the break of the attachment between these proteins. And so one of the mechanisms we will see in preventing the release of the virus is by inhibiting those enzymes that facilitate the exit of the protein, as we will see in the life cycle. Okay? Yes, Eric?
For the I see, I noticed the little green stuff, right, for the envelope. Is that something that's naturally made because the virus has already incorporated its DNA into the nucleus? It's already made, right?
Excellent question. So, to be honest, I'm not sure if it's present in the living cells already or if they exist because of the virus. But I know one of the cases, like the H1N1 virus, it has its own neuraminidase, it's called, enzyme that is used to facilitate the exiting of the virus. So my guess is coming from the virus itself. Okay?
Thank you.
Sure. Any other questions? Okay. These are some examples on DNA RNA viruses. So the name of the virus and the name of the disease. Not for my, portion. I would not ask you questions about tables like that. But again, you might want to know that for the pharmacotherapeutics. Okay, let's talk about drugs now, antiviral drugs. So we've learned about viruses in general, the life cycle. Now drugs will be targeting all these steps in the life cycle, okay? So, of course, antiviral drugs are a class of medications used for viral infection. They do not destroy the virus itself, they just inhibit its development. So unlike the case with the bacteria and fungi, where some of the antibiotics breaks the bacteria or kills it, or the fungus, in the virus, the virus itself is not a living organism. It's just a bunch of proteins and DNAs. So to stop the virus spread, we just prevent its life cycle. We just, I guess, inhibit its production. Okay, right. And this is just to say that the first antiviral drug discovered in 1963 is the idoxuridine for the treatment of the herpes simplex. Okay, so let's talk about the antiviral drugs. There are 6 classes of antiviral drugs. Okay, so as you can imagine from the life cycle we've studied, we would see agents that inhibit the DNA polymerase. Now, we've said that we have viruses that are DNAs, and so those DNA viruses, they have a protein called DNA polymerase that is responsible for generating multiples of those DNAs. So single DNA for the virus, DNA polymerase would actually be responsible for generating multiple DNAs. Okay, so one way to stop the spread of the virus is by stopping the replication of the DNA. So you would design DNA polymerase inhibitors. Okay, now in the case of the RNA viruses, we've said there is an extra step needed to convert the RNA into a DNA. And we talked about an enzyme called transcriptase, and there is reverse transcriptase. So the enzyme transcriptase is the one that we learned in protein synthesis. It converts mRNA into, DNA into mRNA. The reverse transcriptase converts the mRNA into DNA. Why do we need this step? Why is this step essential?
In terms of viruses, right? Is that what you were saying?
Yes.
Oh, yeah, it's because it needs to be converted into DNA so that way it can get incorporated.
Excellent. It needs to be converted to the DNA such that this piece of DNA could be then inserted into the human or the living cell DNA. Okay? Okay, so to inhibit the life cycle of the RNA viruses, you could actually prevent this reverse transcriptase. Okay? And then we have 2 kinds of these reverse transcriptase inhibitors. We have things that are similar to nucleic acids, and so we call them nucleoside reverse transcriptase inhibitors and RTIs. And then we have things that inhibit the enzyme, but they do not look like nucleic acid. And we will talk about why do they mimic the nucleic acid or not. Okay, so we have non-nucleoside reverse transcriptase inhibitors. Now this acronym is actually very common. Please make sure you catch that. So many times they will refer to drugs, antivirals, as NRTIs or NNRTIs. Okay, so just make sure you catch this acronym or this term. Okay. All right. So, but then we also have drugs that interfere with the cellular penetration and early replication. So the fusion of the cell, the entry of the virus into the cell, we have drugs that inhibit that. Okay. And then let's go to number 5 because it's related. We have fusion inhibitors. Okay. So there is fusion and there is penetration. So there are drugs that could prevent the fusion, and then there are drugs that once the virus is inside, it has to uncoat, penetrate. So there are drugs that inhibit the uncoating. So I think this figure here should help. Okay, so there is fusion. Okay, and then once it's inside, there is something that prevents the penetration. Okay. We also talked about, of course, DNA polymerase inhibitors and RNA reverse transcriptase inhibitor. Okay, and then we also have neuraminidase inhibitors. Okay, so what is neuraminidase? When the virus is ready to leave the cell, as it's forming this budding, I guess, this sphere will be attached to the surface by something called I can't remember it now. It's somewhere. We'll get to that when we talk about neuraminidase inhibitor. So this enzyme is needed to break this link and allow the virus to leave. Okay? So by inhibiting this enzyme, the virus will not be able to leave and infect the next cell. So these are neuraminidase inhibitors. Okay? Right. And then we have something called, let's get to the integrase inhibitors. Remember when we said DNA will has to be inserted into the DNA of the host, right? So DNA of the virus has to be inserted. So there are enzymes or proteins that are responsible for doing that. It's called integrase enzymes. Okay, so those integrase, they integrate the DNA of the virus into the DNA of the host. Okay, of course, in the case of the RNA virus, we talked about the extra step where the RNA has to be converted into the DNA and then DNA will be inserted. So the enzyme responsible for inserting those viral DNA is called integrase, and so there is an inhibitor to that. Lastly, there is something called protease inhibitors. Okay, so at some point once the DNA is inserted into the host DNA, the host will start producing proteins. Okay, now this protein is actually a single chain. It's a large single chain of protein that's only specific for the virus. This protein is inactive. It has to be chopped into all the proteins that are needed by the virus. Okay? The process of cutting this protein is performed by an enzyme called protease. You might think about the name, it's proteinase, it's something that cuts the proteins, so protease. And so we have protease inhibitors. Make sense? So that's the last mechanism. So we have 6 mechanisms that we'll visit today. Any questions about the mechanisms of antivirals? Yes.
All these enzymes are enzymes from the virus, correct?
Excellent. So all these enzymes are enzymes from the virus. However, we do have similar enzymes in our body. For example, we do have DNA polymerase, right? So there is some similarity between them. And so the goal is to design something that's specific to the viral protein, but not to the human protein. So there are some changes. If we could be careful in designing something that attacks only the viral, then we'll be in good shape, okay? The same thing with the protease. We do have protease enzymes in our body, right? You might remember that. We do have protease, right? You remember that? So there is protease, viral protease, which actually it's actually symmetric. It's like one half and the other half are symmetric, unlike the human body. So you could actually take advantage of that and like design something that only specific to the viral protease. Okay. Integrase fusion. Do we have a fusion? Well, a fusion is these are like some receptors at the surface. Integrase. I can't think of integrase enzymes if we have in our body or not, to be honest. Because I can't think of why would we need to integrate that. Yep. You had a question too, Eric?
Yeah, just 2 questions. The first thing is that like when you're talking about like the neuroamidase ones, right? We learned at least in the previous most that zanamivir and sotavir is anything related with like I believe the influenza virus. So
Yes.
is
True.
this only for the influenza, or could this be for like any kind of systemic viruses?
Excellent. So I'm not sure if these are present only in the influenza. I know, like you said, for sure they are in the influenza. In fact, that's why the H1N1, you've guys heard of the H1N1 virus? Okay, so the H stands for the type of the hemagglutinin protein that's helping the adhesion, the fusion of the virus. The N stands for the neuraminidase enzyme. So we have hemagglutinin type 1 and neuraminidase type 1, and that's what the H1N1 virus. So Yes, it's of course, you know, one type of the influenza virus, but I'm not sure if it's only present in the flu and the influenza virus or not. I'm not sure if it exists in other viruses too. Yes.
This is another question too, right? Like when you mentioned that there's like something that's so different from like viral, like you know, from viral components versus like actual human components, and we want to make like careful formulations that doesn't attack humans. How could we really be able to tell between a viral and a human? Like, just like, how do we incorporate that?
Excellent. So the question is, how can we be sure that, you know, we have enzymes or proteins in our body that mimics them? We actually would investigate the binding site. We look at the binding site for the human enzyme and look at the binding site for the viral enzyme. And you might see some amino acids that are different, and then we would take advantage of that. There's something called docking, where you dock the drug into its binding site. So we could perform docking studies to see which drugs could be binding selectively to this, but not to that, okay? And then filter those out and use them,
Okay.
okay?
Thank you.
Sure. Any other questions? So far so good? You're having fun?
Yes.
All right. Okay, let's start by the first one, which are the ones that inhibit the DNA polymerase, okay? This is the list of DNA polymerase inhibitors. You have a drug name, you have the brand name, the spectrum of activity, and then some dosage forms. I don't expect you to memorize any of that, of course, but you will need that for the for the therapeutics. But this is just for your information. Now we will cover some of them, actually most of them. So, but they all follow the same mechanism, okay? All right, so the first one of the DNA polymerase inhibitors, these are the structures again. As you can tell, well, let's get to them and then you will see what I mean. So the first one is something called acyclovir, okay? So, first of all, let me remind you, so these are DNA polymerase inhibitors, okay? So they prevent the multiplication of the DNA. How does the DNA get multiplied? You will have split of the DNA, and then each strand, you will have nucleic acids being added, okay? And so these drugs, polymerase inhibitors, they use a mechanism called false substrate mechanism. What does that mean? So these drugs, they mimic the nucleic acid, okay? And then they fool the DNA polymerase. DNA polymerase thinks that, this is a nucleic acid I should incorporate. Once they do that, this drug is usually missing the link to the next nucleic acid, and so it causes chain termination. Make sense? Everybody get that? So the mechanism they rely on, it's called false substrate. So the drugs, if you look at their structures, they actually mimic nucleic acids. I'm not sure if you remember in your organic chemistry, in your biochemistry. So nucleic acids would have the sugar unit, and then the nucleic acid, nucleotide. So then you would see that in all of them, okay? And so they mimic, in this case, they mimic guanosine, nucleic acid, okay? And so it gets incorporated into the DNA thinking that it's a guanosine, but then usually it lacks something, it's lacking the OH group or some kind of groups that are necessary to link the next one. And so it would block the growth of the DNA, and it's called chain termination. Make sense, guys? Now the same mechanism we would see with the reverse transcriptase inhibitors that are nucleoside. So remember we talked about the reverse transcriptase? We have two kinds, right? We have nucleoside reverse transcriptase. They have exact same mechanism, the false substrates, okay? Non-nucleoside is something else. They just competitive inhibitors. All right, so let's look at this acyclovir. So that's the structure, okay, of the acyclovir. Actually, that's after they add the phosphate. So just wait on the phosphate first. So acyclovir is a guanosine nucleotide derivative. It's sufficiently similar to the normal nucleoside so that it could serve as a false substrate, I should say, for the DNA polymerase. Since it lacks the sugar, it lacks the 3' hydroxyl group needed for the sugar, then it would cause chain termination. Okay. Now, what's interesting about this acyclovir is that so this is the acyclovir. Okay. What's interesting is that it has to be phosphorylated 3 times. First phosphorus group, a second, and then third before it could be activated. So would be an example of a prodrug. Why? Because by itself, it's inactive. It's not able to inhibit the, it's not able to cause chain termination. It has to be phosphorylated to have, 3 phosphorus groups before it could act as a chain terminator. Make sense? And so it has these enzymes, HSV1K, GMP kinase, NDP kinase, to be converted into the active form. Okay? Make sense? And so what's interesting is that the first step in the phosphorylation can be carried out only by the viral enzyme. It's called viral thymidine kinase. Viral thymidine kinase. Okay? Why is this important? So this step cannot be carried out by the living, the host enzymes, only by the enzyme. Why is this essential? Why is this advantageous? Yes, Eric.
Because didn't we say that we wanted to have something that's more targeting specifically with like the viruses versus something that's more human-like?
Exactly. That's going to help us with selectivity and specificity. Okay. So, do you agree with me that only the cells that are infected by the virus will be the ones that will be able to activate this drug, because they will have this enzyme, right? So the normal cells will not have the viral enzyme, so they will, and the cellular enzyme is not able to convert it, so it's not going to be activated. So that's very nice, because you could actually make sure that the drug is selective. It's only being activated where it needs to be activated, which is in the cells that are infected by the virus. Make sense? Everybody get that, guys? So if I ask you what's the mechanism of selectivity for this drug, what would you say? Or if you have choices for mechanism of selectivity for this drug, what would you say? Yes, Eric.
It is specifically able to go and work on toward like viral thymidine kinases, and being able to go and target that is essential because this specific medication is able to go and only attack that viral version.
Right. So, but just to be clear, it's not targeting, it's being activated by the viral enzyme. So by being activated, this drug will only be functional in the cells, host cells that have the virus, okay? So they get converted into the first phosphorylated form, and then the next could be converted using the cellular enzymes, so it would be phosphorylated the second time, and then again cellular enzymes phosphorylated the third time, and now it's in the active form. Okay, before that it was a prodrug, now it's in the active form, and then it could now cause the DNA termination, cell termination. Make sense? Okay. Let's see, it has potent activity against DNA viruses including HSV1, HSV2, so these are again, pharmacotherapeutic information. I'm sure you're gonna need to know that as far as the spectrum of activity. Pharmacokinetics, the drug is administered by IV. It could be taken orally, but it does not have like the best oral bioavailability. We will see that that could make some modification to enhance its oral availability. Okay, and could also be used topically. Okay, it distributes very well to the cerebrospinal fluid. That's very nice. So if you have viral infections in the cerebrospinal, you could use that. Excreted in the urine, and so it would be affected by, well, through glomerular filtration and tubular secretion, and so it could be affected in case of renal failure. I think we talked about this in the past. If you have renal failure versus hepatic failure, you want to pay attention where the drug is being excreted or metabolized. In this case, if somebody has renal failure because the drug is primarily excreted into the kidney, then you would avoid it. Okay. Now, the drug itself could actually cause renal dysfunction at higher doses. Okay. So at higher dose of this drug, you might cause renal dysfunction, and then, of course, the drug would even accumulate more. Side effects depend on the route of administration. For example, local irritation in the topical, headache, diarrhea, nausea, vomiting in the oral administration. Resistance. So generally speaking, with viruses, bacteria, and fungi, one of the ways for having resistance is if you think about the target or the enzyme that's been affected. This enzyme could have a mutation, and now it would result into something that could bypass this drug and not affected by this drug. Okay, so in this case, the DNA polymerase or some viruses will be deficient of the thymidine kinase, which is necessary for the activation of this drug. So that's one of the ways of mechanisms. So you could have different viruses that either do not have the thymidine kinase needed to activate the drug, and so the drug will never be activated, or you may have different versions of the DNA polymerase that are more selective, and so they do not get fooled by the false substrate. Okay. And then cross-resistance to other agents in the family occurs. So if a drug, what does that mean, cross-resistance to other family occur? Yes, Eric?
I think that means that if one specific viral host cell has a resistance, it's able to go and share that DNA piece with other specific viral.
Oh, so actually, within the same family of DNA polymerase inhibitors, if a virus is resistant to one drug within that family, there's a good chance that this virus will be also resistant to other drugs in the same family. Okay, so we have DNA polymerase inhibitors. If we have a virus that's resistant to acyclovir, then we should be careful. We should avoid the entire family because there is a chance that this virus is resistant not only to this drug but to all the DNA polymerase inhibitors. Make sense? So that's the cross resistance to other drugs in the family. Okay, now we've said that this drug could be taken orally, but the oral bioavailability is not the best. We could enhance that. So there is something called val-acyclovir. So it's acyclovir, but you add a val because we're adding actually valine here. Let me just enlarge this structure. So we have added a valine, which is an amino acid, by the way. We have an added valine structure to the drug, and so we have enhanced its oral availability, and we came up and we'll have now something called val-acyclovir. Okay, so it has much higher oral bioavailability. Okay, and then of course this valine could be broken down in the intestine or the liver after oral administration and be converted into the acyclovir to be active. So this is actually prodrug. Okay. Everybody get that? So what's the purpose of designing the val-acyclovir? What's the purpose of doing that, and how did we do it? So yeah, Eric.
We added the valio ester on the acyclovir section.
Valine amino acid. Yeah.
Yeah, and then what we did is that this is essentially a prodrug that basically has a higher oral bioavailability.
Excellent. This is a product that has higher bioavailability. Once it crosses the intestine and the liver and makes it to the circulation, it will be converted into the acyclovir. Okay? Everybody get that? Questions? Yes, Eric.
You mentioned like like the renal dysfunction with the cyclovir, right?
Yes.
I'm not sure if this is true, but one of the counseling points then would be to like tell the patient to drink lots of water at the time when they're taking medication.
That's a good idea. I'm not sure if, I mean, rationally thinking that makes sense, but something that we will learn is that we use our basic understanding and knowledge to rationalize things and then design hypotheses. And then we could test them clinically to see if actually it's significant and you could see the value of that and the effect. I guess our body is very complicated. We can make hypotheses based on our rational understanding, but then we have always to confirm it with clinical studies. Okay, so to me, this makes sense. Drinking plenty of fluids because it might cause, renal problems, but we have to check that clinical studies and see that it actually does make a difference. Okay,
All right, thank you.
sure. Any other questions? Oh, it's 1:56. I'm sorry. I didn't realize that. So let's have a break before, let's see what's next. Okay, before we start with the next drug, let's have 10 minutes break and come back at 2:05, okay?
How are you? I have a few questions. So I wasn't understanding what's the mechanism of selectivity.
All right, let's continue. Okay, so far we've been talking about the DNA polymerase inhibitors. We've seen the acyclovir and the GAN acyclovir. The next example is actually very similar to the acyclovir and GAN cyclovir example. We have something called, we had acyclovir and val acyclovir. Okay, so now the next example is we have the GAN cyclovir, and then we have something called val GAN cyclovir. Exact same concepts, nothing new. Okay, we have, of course, structurally different drug called GAN cyclovir. Okay, and unlike the acyclovir, it's not available. It cannot be taken orally. So we have to modify it and add a valine amino acid to make it available orally, and so you would get the val GAN cyclovir. Okay, the rest are just some information about the like their application, their therapeutics. Like I said, same mechanisms, same inhibition. This is available by IV. It could cross the blood-brain barrier to the CSF. It's not available orally. It has to be administered into this form to be taken orally. Okay. Same example with the resistance. Adverse effect: carcinogenic, embryotoxic, teratogenic, and so forth. It could cause neutropenia also, depending on the dose. Any questions about this example? So what does this unit here mimics? Which nucleic acid? Guanosine. Thank you so much. So one of the things I want you to know is to differentiate which amino acid it mimics. Most of them mimics guanosine. Okay, if you look at the structure, most of them have this guanosine unit here. Guanosine, guanosine, guanosine. Well, actually, this has to be oxidized, but still, guanosine, except this one here, cytosine. We'll get to that soon, okay? Just to help you memorize, it's called cytovir, so the cy is from the cytosine, okay? Any questions? So this one here is straightforward example that just the same ideas applied again with the acyclovir. Okay, the next one is something called pencyclovir. Again, it's a guanosine analog. Okay. We also have a prodrug for this one. It's called famciclovir. Okay. So we have the pencyclovir and then we have the famciclovir. So first of all, the pencyclovir, again, it's a guanosine nucleotide analog, active against HSV1, HSV2, VZV. It's only available topically. Okay, so the pencyclovir can only be administered topically. It cannot be taken by IV or orally. Okay, and just like we've seen with the other ones, it has to be monophosphorylated by the viral enzyme, which is helpful for selectivity. And then the active form has intracellular half-life much longer than the acyclovir, so duration of action is longer. It's negligibly absorbed when taken topically, which is actually true for most of the drugs. When you apply something topically, you only get negligible amount of the drug gets to the circulation, which is nice. So you could avoid, side effects. And then we have the prodrug of this one. If you pay close attention, you would see that they have added these ester groups, okay, to mask the OH groups here, and so the drug could be taken orally. Okay, and then of course once it's inside the body, it gets activated into the active form. So it's a prodrug of the pencyclovir. It's called famciclovir. Okay, and these are, and so now it could be taken orally. So these have activity on herpes zoster, genital HSV, and herpes labialis. Adverse effect: nausea, headache, and nausea. Any questions about the pencyclovir and famciclovir? Yes.
So technically, all the cyclovirs, both of them would be prodrugs because the tablet
excellent point. Yes. So even though you've designed, so repeat her question. So she's saying that for the cyclovirs or these DNA polymerase, both forms are actually prodrugs. This is a prodrug, and this is also a prodrug. That's true, because even when you convert this into the the drug that we want, this still has to be activated with the viral enzyme. Okay? So it's a prodrug of a prodrug, I guess. Yes. Okay? All right. Next one is cidofovir. This is the one I pointed out that it's an analog for the cytosine, not the guanosine. Just pay attention to that, okay? You could tell from the structure. This is the structure. If you look at the structure of the cytosine nucleic acid, you would see that it has this cytosine. Okay. What else? Oh, one thing about the cidofovir is that it actually already has the first phosphorus in the structure already. Okay, so is that a good or bad thing? What do you think? It's good? What do you think? Yes, the first step is a viral specific, so that shouldn't be, it should not be good, okay? Because we rely on this for selectivity, okay? So this drug already has the first phosphorus already in the molecule, so it would not be as selective, okay? These are some like therapeutic applications for the drug. It's administered by IV. It's also given by intravitreal injection. So this is in like the cornea of the eye. In the case of the, I guess eye infection. It has significant renal toxicity. Okay. So notice that most of the time when we talk about these polymerase, we talk about the renal toxicity as something common. It's contraindicated, of course, in renal impairment, obviously, okay, or to be taken with drugs that could also cause renal toxicity. So if you have drugs that already could cause renal toxicity, and this one also can cause renal toxicity, so obviously you shouldn't administer them together, okay. Now, one way to enhance or reduce the renal toxicity effect is by administering something called probenecid. Probenecid is a drug that inhibits the transport or excretion of the drug from the kidney into the urine. So it blocks the elimination of the drug into the urine. As a result, this drug that's administered with probenecid will accumulate. And so you could combine probenecid with some drugs and then administer smaller dosages of those drugs. And this would reduce the toxic effect of this drug. Everybody get that? So if you have a drug that could cause toxicity, okay, in this case it's causing nephrotoxicity, and so instead of giving it in high doses, you could give it in small doses and then prevent its excretion using probenecid. And so this way you could give smaller dosages of the drug to protect prevent the toxicity. Does that make sense, guys? In fact, this example is also common with penicillin drugs. With the penicillin, to prevent giving higher dose of the penicillin, you could give it probenecid with it, and then you could give smaller amount of the penicillin in this case. Okay? Does that make sense? Yes, Eric?
Like, I'm just curious, right? Like, if it is to be eliminated, right, renally, without the probenecid versus the one with it, like, do you know, like, what's, like, the, how do I say it? like a time window difference between if you do give it with propenicid versus when you do? Like in terms of elimination half-life, how it eliminates the mind.
The time difference between like the time you administer these two, or like the time.
Like when you administer them both at the same time,
Mhm.
one with the propenicid and one without it.
Okay.
Would there be like a difference between like the time that it takes for it to actually go and clear out? And if so, do you know like what's the time difference with that?
For elimination.
Yeah.
I'm not sure about that. So the question was, if you have two cases, one with propensid, one without, what's the time difference for the elimination when it starts the drug getting eliminated with the propensid or without? I'm not sure about that. Yeah, but I guess I guess the take-home message is the point of having propensid is the drug would be less eliminated, and so you don't have to give as much of the drug, and the concentration of the drug in the plasma would be much less, and so the toxic effect of the drug would be much less. Okay?
Thank you.
Sure, absolutely. Any questions so far? Okay. Right. Lastly, we have something called foscarnet. What's unique about this drug? How is it different from all the ones we've seen? This is another DNA polymerase inhibitor. Yes.
This one doesn't have like the guanosine kind of ring.
Exactly. It doesn't have a nucleic acid at all. So the ones we've seen, most of them had guanosine, and then we had cidovir, which has cytosine. Okay, cidofovir, sorry, has cytosine. This one, foscarnet, it's a phosphate from the name phos. Foscarnet, it's a phosphate. Okay, so no nucleic acid. So this is not mimicking nucleic acid. How does it act? How does it work? This actually competes with the site where the phosphorus or the phosphoric groups bind to the DNA polymerase, and so it interferes with the job of the DNA polymerase and blocks it. Okay, so it's more like a competitive inhibitor because it mimics the phosphorus groups. So when it's time for the DNA polymerase to implement those nucleotides with their phosphorus group, this interferes with where the phosphate binds, and then it blocks the DNA polymerase. Okay, and it's selective to the viral DNA. Okay, it's not affecting the human, and the mechanism of selectivity is actually based on the concentrations. What does that mean? How is the concentration here helping us with the selectivity? So let me give you a hint. So at smaller concentrations, this drug would inhibit or interfere with the viral DNA polymerase. But at higher concentrations, it would inhibit both DNA and viral. So you could administer certain slow enough, just right amount of the drug to interfere with the viral DNA polymerase, but not the human. Okay? I guess I give the answer, not the hint. So but I guess the question is, why is that true? When the living cell is infected by the virus, it's actually constantly and in high speed producing all these viruses constantly. So the DNA polymerase is consuming the phosphorus and nucleosides in a much higher rate. So the need for the phosphorus is higher. And so you could give like smaller concentration of this drug, and it's going to have higher impact on the cells that are rapidly, using this enzyme. So there's a higher need, and so they will be impacted more by this drug. Does that make sense? So you're relying on the fact that these cells are constantly functioning and using the DNA polymerase, and so the need for this phosphate is higher, and this would interfere with the phosphate group, and then the effect at smaller concentrations would be more than the effect at smaller concentrations at the infected cells would be more, and the normal cells. Make sense, guys? No?
Could you mind explaining that again?
Okay. Think of the cancerous cells. They're dividing at a higher rate, okay? And so they're consuming, I guess, or they're using the enzymes at a higher rate. So if you interfere, if you administer a drug that interferes with those enzymes, but you administer it at certain concentration, so the need for these drugs at those cells dividing constantly or at a higher rate is going to be more. So they'll be more impacted by the drug than cells that are dividing at normal rate.
Okay, so basically it's just that the more it's able to, like, you know, build up more of the viruses, that's basically meaning that we need more concentrations.
Yes.
Okay.
So because it relies more, because there is more need and demand for this phosphate, this drug is going to interfere with the phosphate and affects the cells that has the virus compared to cells that don't.
Oh, so it's more selectivity.
It's more selectivity in terms of concentration.
Okay.
So there is more need for the ones that are dividing because of they have the virus, they're affected by the virus, than the normal ones. And so they're more impacted by the drug at certain concentration, okay?
Okay.
Yes.
Just confirming, because there's no nucleic acid, right? It's not a false drug.
Excellent. Yes. So that this drug doesn't have a nucleic acid. It's non-nucleotide analog. Non-nucleoside analog. Okay. It is not correct. It is not a false substrate. Okay. Absolutely. It actually binds to. It just binds to the site where the phosphorus group should be binding to, interfere with them, and blocks the DNA polymerase, okay? So it's reversible, inhibiting to the viral DNA and RNA polymerase, okay? Interfering with the synthesis. Why? Because they mimic the phosphate, and so they selectively inhibit the pyrophosphate binding site on the DNA polymerase. It's like a competitive inhibitor. In fact, the same concept you will see with the reverse transcriptase inhibitors when we have nucleoside and non-nucleoside. So this is the same concept. You would see things that mimic nucleosides and then act as false substrates, and you see drugs that don't mimic nucleosides. They just go to the binding site and interfere with it and inhibit it, okay? And just remember the phos for the name, and that the fact it's from the structure. I mean, usually all the structures will be provided, so you won't have to, you can tell from the structure that it's a non-nucleoside, okay? And applications, of course, you're going to learn that later. The same thing with the resistance. Mutations to the polymerase structure could produce resistant DNA polymerases. Any questions? Excellent. Okay. Pharmacokinetic of the phoscarnet, poorly absorbed orally, so it must be injected by IV. Should also be given frequently to avoid relapse when plasma level falls. So should be constantly administered because it's eliminated quickly. It dispersed throughout the body. 10% goes to the bone matrix. Let's see. The parent drug is eliminated by glomerular filtration through biosecretion. Nephrotoxicity is a common adverse effect. Of course, there's anemia, nausea, fever. Now, among the side effects also, because it's phosphate and negatively charged, it could bind to positively charged cations such as calcium and magnesium, and so it would cause hypokalemia and hypomagnesia, which makes sense, right? So the phoscarnet is negatively charged. It binds to the calcium, magnesium, which are positively charged, and then deplete them. So you would have hypocalcemia, hypomagnesia. There's also hypokalemia and hyper. So it interferes also with the phosphate level, and then it, as a result, causes seizure and changes to the heartbeat. So anytime you interfere with the potassium level, hypokalemia, then you might remember the sodium-potassium pump in the nerve pulses. So interfering with the potassium could affect the the neurons and also your heart muscle. So you have arrhythmias, and then also you may have seizure. Make sense? So far, so good?
Dr. Khan,
Yes.
you mentioned like hypo
Honestly, okay, so the question is, we've mentioned hypo and hyper. So, I mean, I'm not sure which one, I mean, why are they both listed? I mean, I could guess. I may not know the exact answer, to be honest. I'm sorry. I mean, I'm not sure if because it's in some areas it might cause hypophosphatemia because it mimics the phosphate, so you don't have the phosphate. In other areas, it might act as the phosphate, but so then it's like having hyperphosphatemia. So depending on how is it this drug is treated, like is it treated as a like if it's recognized as a phosphate, and then you will have so much phosphate, or maybe some areas or some enzymes might think that it's not phosphate, and so you would have hypo.
Okay, got
So
it.
this is just my guess. Okay,
Yeah, okay.
sure. So far so good. Okay, so the next class is going to be much easier because now we understand the false substrate. Okay, so we've talked about the reverse transcriptase. We said this is the extra step needed for the RNA viruses. So remember, this class would only work for the RNA viruses, right? This would work for both DNA and RNA viruses. Make sense, guys? This step, reverse transcriptase, is needed for the RNA viruses because they convert the RNA into the DNA and then it gets inserted. Okay, so we have two kinds, a nucleoside. Now you should know what we mean by that. There are nucleoside inhibitors, so they mimic nucleosides, and then we have non-nucleoside inhibitors. Let's look at them. Okay, so these are the list of drugs again. So generic name, brand name, the common name, and then dosage forms, which again, this is just for your information. And these are the structures. So we have things that have nucleoside, okay. These are the nucleoside reverse transcriptase inhibitors, okay. And then we would see next the non-nucleoside. To start with, we have zidovudine, okay. So it actually mimics the thymidine, okay. So it mimics the deoxythymidine analog, okay. So this is the thymidine, deoxythymidine, it mimics that. And the same concept, it actually has to be phosphorylated. The first step, second and third to be converted into the active form, and then causes chain termination to the DNA. Okay. Remember, the reverse transcriptase is converting RNAs into DNA, and so we're building this DNA, and then right in some point where we add this drug, it's gonna cause the chain termination. Okay. Let's see. These are some uses for the drug, and then the side effects: myelosuppression, anemia, neutropenia, GI tolerance, headache, insomnia, and so forth. Any questions about the zidovudine? Any questions? Yes.
And you're saying that, like, okay, like these medications, I just wanted to make sure, the medications for the nucleoside reversible and the other ones that we talked about, these are only for like RNAs.
Yes. So why is that? Because that's the RNA virus are the ones that are going to need this step to convert the RNA into DNA and then insert it into the host DNA. The DNA viruses, they won't need this step because they already have DNA. They get it inserted into DNA and then they continue with the life cycle.
Okay.
Okay?
All right, thanks.
Sure. Questions? All right. You guys are going to fall asleep soon. Okay. Next example is called ribavirin. Okay, the same mechanism. Let's see. The only important thing I want to point out is that it's contraindicated in case of pregnancy. Okay. Applications, you're going to learn that in the therapeutics for hepatitis C. It's used in combination with interferon, which is we're gonna study soon actually. as an antiviral. Broad spectrum against RNA, DNA viruses. Synthetic guanosine and therefore that must be interfered with. I have a question mark about the DNA viruses here because we just said they act only on the RNA. So let me get back to you on this, okay? Pharmacokinetics, effective orally and by inhalation, eliminated in the urine, contraindicated in pregnancy. I think this would be something important I really want you to know, okay? The fact that it's contraindicated in pregnancy. Any questions? So we only talked about 2 examples in this nucleoside reverse transcriptase. Any questions about them? Okay, the next class are the non-nucleoside. So we talked about this, now we're going to talk about this. Okay. These are the examples. I mean, from the structure, you can tell that they do not mimic the nucleoside, right? So again, these are the tables just for your information. From the structure, these are ones that competitively inhibit the reverse transcriptase enzyme. They compete with the binding site and then block it and inhibit the enzyme, okay? They do not mimic the the nucleosides. We just talked about the mechanism of action. There is clinical application. There is some side effects. Nothing unique. Any questions? Okay, so you should be able to look at the structure and tell that this is a nucleoside reverse transcriptase or non-nucleoside reverse transcriptase. Make sense? Fair enough? Okay. All right, so now we covered all these. Let's talk about the next ones. So we're going to talk about the antiviral agents that interfere with the cell penetration and early replication. Okay, so we're going to talk about things that interfere here. Okay, so I think the first example is the amantadine and ranitidine. Sorry, the amantadine and rimantadine. Yep. Okay, so these are the examples of drugs that interfere with the cellular penetration. Okay. Again, this table is for your information. The first example is the amantadine. It inhibits the penetration of the RNA viral particles into the host cell. How is that happening? By blocking the uncoating. Okay, so apparently, so when the viral enters the cell, it's coated by this vesicle here. Okay, and there is a proton pump, here called the M2, matrix 2. This pump is responsible for pumping protons inside this vesicle. And as a result, the pH inside the vesicle would change, and this vesicle will break. Okay? So there will be like a pH shift into the too much acidic, so the vesicle would break. How? By the act of this pump that inserts the potassium inside. And so guess what? These two drugs, they could block the pump, the matrix 2, and then prevent the pH change, and then there would be no break of that vesicle, and the virus could not penetrate the cell. Everybody get that? Okay? All right. So one thing you might consider is, since this is a pump that's acting on the proton, which is positively charged, the drug itself, you would guess that should have a positive charge on it. And that's where the importance of the nitrogen groups come in. So you might remember the ionization, and ionization of the groups. You remember that? Different forms of ionization and ionization. So this nitrogen will be ionized into the NH3+, the same thing with this, okay? And this will compete with the proton at the pump, and then it's going to prevent its function, So that's their mechanism of action. Okay, at this matrix 2. And so if you substitute any of those any of this nitrogen with something like OH, thiol, or cyanide, it's not going to work. You're going to get inactive form of the drug. Okay. Now, just so you know, this structure here is called adamantine, and then by substituting this R with the nitrogen or with the nitrogen and methyl, then you get the amantadine and rimantadine. Make sense? So everybody get that? So we talked about mechanism, should make sense, how it inhibits the matrix M2 pump from pumping protons, it blocks the pump, and then we also talked about the importance of the nitrogen. If you substitute it with any of these, any groups really, it's going to become inactive. Okay? So far so good? All right. Clinical applications, type A strain of influenza virus, type A strain of Asian influenza virus, and it could also use for Parkinsonism. So amantadine is one of these drugs that has a polypharmacology feature. It could be used for antivirus and then for Parkinson's as well. Side effects, nervousness, confusion, headache, drowsiness, insomnia, depression, nausea, diarrhea, hallucination, constipation, anorexia. Not bad. I would deal with the virus much easier. Okay, so this class of drugs that interfere with the penetration, amantadine, rimantadine. Now we have a different class of drug that also interfere with the penetration. These are the interferon alpha. Okay, so to start with, when our body is infected by viruses, they have their own defense mechanism. So they actually produce interferons that interfere with the penetration, uncoating, as well as even the virus replication. Okay, but these might not be enough, obviously, because we'll have, otherwise we wouldn't have the infection. And so one way to take advantage of that and then to fight the viral infection is by administering interferons. Okay, you could actually genetically synthesize interferons, which are the proteins anyway. So these proteins would be synthesized genetically and then administered to our body to help fight this virus. Okay, and so the interferon alpha 2b is doing this exactly. It's actually interfering with the penetration of the virus and then inhibiting the replication and then inhibiting the uncoating of the virus. Okay. They are used in treating things like hepatitis type B, C, and D. Okay. Now, this interferon, if we want to administer it, of course, it has to be, it's a protein, so can we take it orally? We can't. Proteins can never be taken orally because they will be digested in our stomach. So they have to be taken systemically, like IV, for example. So, but they don't have a long duration of action. So instead of administering the drug by IV more frequently, the interferons would be designed in a way to make them to increase their duration of action by adding something called polyethylene glycol, PEG, and so they are called the pegylated interferons. Okay, what's the job of this polyethylene glycol? It actually prevents the metabolism and excretion of these interferon. It interferes with the how our body eliminates and metabolize those interferons. It prevents that. And so this way you don't have to give the drug as frequent as you would. Okay? Everybody get that? Questions? Yes.
I was thinking, right, like if you add like the polyethylene glycol, I remember like in some rules that we mentioned in medicinal chemistry too.
Yes.
Like, we added more of like, chains. That means that don't we make it also very like
Excellent. Thank you so much. So Eric was saying that one of the rules we use to increase duration of action is by adding lipophilic groups. This way the drugs would stay longer in the body and then have longer duration of action. That is correct. in this case, we're actually using a different mechanism. We're adding to proteins, we would add PEG. You would also see that in other examples, by the way, where they pegylate those proteins such that they are not recognized by the metabolizing enzymes, and they still do their function, but they're not being excreted, metabolized as often, and so you'd increase the duration of action. Make sense?
Yeah, thank you.
Sure. So in short, we have interferons that our body naturally produce, so we could mimic them and design them ourselves, and in order to administer them less frequently, we have pegylated them such that they stay longer in the body. Okay. Ready for the next class? I mean, next category of antivirals? So far, so good?
Yep.
Okay, so we covered this and that, so there's only 4 remaining. Okay, so the next ones are the neuraminidase inhibitor. We already pointed out that mechanism. So there is the attachment. Let me get to the name of the protein now. I could remember it. Okay, there's something called sialic acid, okay, that attaches the viral to the surface of the cell. And so there is an enzyme called neuraminidase, or it's also known as sialidase, okay, that breaks this sialic acid before the viral can leave. Okay, so that's the viral. At the end, before it detaches itself from the cell, there is a sialic acid here that needs to be broken by this enzyme. So you could actually block this enzyme, inhibit it, not block it, inhibit it, and so the virus will be stuck forever with the cell, and it never leaves and doesn't distribute to other cells. Make sense? Get that, everyone? So that's its mechanism of action.
Is it possible you mind repeating that again?
Sure. So when the virus assembled inside our cell, it has to leave the cell. As it's leaving, the last step before it leaves the cell, it's attached to the surface of the living cell by a sialic acid. Okay, and so we have to break this sialic acid using sialase or neuraminidase enzyme, such that the virus can leave. And so if we inhibit this enzyme, we could prevent the detachment of the virus, and the virus would be stuck with the host cell and it does not distribute.
Kind of like a dog on a leash.
Yes.
Okay, thank you.
You're welcome. And then of course you would rely on the immune system to clear it and take care of that. Okay.
Okay, thank you.
Sure. So far so good. We're having fun. Clearly. You're falling asleep. You're lying to me. I prefer the morning sessions, but what can we do? Okay. Next time I should bring with me coffee. Some sugar and coffee. They will get weird. Okay. All right, so that's with the neuraminidase, and something to point, there is another one, the same mechanism, of course. It's called oseltamivir. So we have oseltamivir and we have, we have 2 examples, the oseltamivir and we have the, what's the other one? Oh, we have zanamivir, okay? So both of them are neuraminidase inhibitors. Now this is Tamiflu. You guys, you've seen Tamiflu, right? Tamiflu, it's taken, capsules. This one here is inhaled, it's inhalation, okay? Just like with the Advair Diskus, I'm not sure if you have seen it. It looks exactly like the Advair Diskus. You would like break the capsule and then just inhale it, okay? All right. So we talked about mechanism of action. It helps make symptoms less severe and shortens the recovery. So that's one of the actually therapeutic information with the Tamiflu. What is special thing about Tamiflu? You have to take it at the very early stage of the infection. If you wait, it won't be helpful, okay? So as you can tell from the mechanism, it has to work early before the virus spreads. So the first one, like the first few days of the infection, you have to take the Tamiflu, otherwise it would be too late to have significant effect. Okay, and then once you take it, it would reduce the I guess the time of the infection to 1 to 2 days. There's a list of side effects for your own information. So if I don't point out special things like with the pregnancy, or if I don't say so far we talked about nephrotoxicity, we talked about the pregnancy contraindication, then these are just general information. You don't have to memorize. Pharmacokinetic, of course, oseltamivir taken orally, zanamivir taken by inhalation. It's not active orally, the zanamivir, obviously, that's why it's taken by inhalation. Resistance, of course, you would have different. So which enzyme does this drug work on? Neuraminidase. And so the resistance would be mutations to that enzyme. Okay, makes sense. All right, so check neuraminidase. We talked about that and that. All right, so we have 3 more. It's 2:47. So, and we have, how many we finished? 22 out of 30. So let's cover one more class and then see if you need a break and come back, or we should continue. I'll ask you, just think about it for now, okay? All right, so the next one are protease inhibitors. So remember, once the virus inserts the DNA into the human or the living cell, this cell starts producing the proteins for the viral. However, they do not produce proteins separate. They produce a single chain of protein, like one protein, and this protein includes all the proteins needed. The neuraminidase, the RNA, DNA polymerase, the reverse transcriptase, integrase, all the enzymes that are needed. But we have to chop them, we have to cut them into pieces. And so this protein itself cuts itself. This protease, it cuts itself into all these pieces. And so one way to prevent that is to block the protease enzyme, okay? So if you look at the chain of the proteins and where it cut, okay, we could have drugs that interfere with where the protein cuts. These are all the list of the drugs that are protease inhibitors, okay? It's one of the most efficient ways actually for preventing the viral infection. You guys remember the coronavirus? What was the drug that's taken orally for that? Paxlovid. What's the mechanism of action for Paxlovid? Protease inhibitor. Okay. So it's one of the easiest way. I mean, not easy, but that's one of the more common way to design antivirals. Okay. In fact, I remember working on one of the projects. I had a student that was interested before they discovered the oral Paxlovid, but Pfizer beat me to it. Now you're laughing. Okay, good. Some of you are awake. Okay. Anyway, so now what's interesting is that when you have an enzyme that protease that cuts protein, so one way to design a drug to inhibit the enzyme is by looking at where it cuts and then design a drug that mimics it. Make sense? So if you have a chain of protein and then you have protease that comes and cut it, so the way that is used to design drugs is by looking at the place where this protein is being cut. Usually it's amino acids, a list of amino acids. And then you would design a drug that mimics that part, that piece, such that the protease will get confused and attack the drug instead, and the drug would bind and blocks it forever. Make sense? Or not forever, but blocks it either reversibly or irreversibly. Make sense? And so this is what we're trying to explain here. So we have the structure of the 2 amino acids where the protease cut, okay? And then they design a drug that mimics that, okay? I'm not sure you can see how similar they are. So you would have some similarity here, here, and here, okay? So they would design this drug that mimic the place where the protein cuts, and then the drug would bind to the protein and then blocks it. So that the same concept was applied in all these protease inhibitors. Okay. So here's the list again: drugs, names, brand name, dosages. These are the drugs. I'm not sure if you can tell how similar they are to the peptide. So you might remember the protein or peptide is the list of amino acids connected together with a peptide or amide bond, right? So if you check, you would see that there's an amide bond here, amide bond here, amide bond here. So they're trying to mimic the protein that's being cut such that the enzyme gets, fooled and then attacks it instead. That's design. So that's the design mechanism. Okay, so the first example is something called saquinavir. It's the first protease inhibitor approved by the FDA in 1995. It's a carboxamide derivative, meaning that it has the carboxamide group, if I can see it here. Carboxamide. I didn't see it, but I know you don't care, so it's okay. Right. Mechanism of action, as we explained, it mimics this cis bond. Okay. So that's the bond that actually gets cut by the protease. Okay. It contains the hydroxy ethylamine moiety rather than this phenylalanine and proline amino acids. So the cis bond is the bond where the protease identify to cut between the two amino acids. So this moiety mimics that, and then this is where the protein gets inhibited, I guess, by the drug. Okay. Clinical applications, side effects. Any questions about saquinavir or in general protease inhibitors so far? Does it make sense, guys, what we talk about so far? Yes, Eric.
So you mentioned that like the nitrogen sections is kind of what mimics that, that pro-cis bond, right? The nitrogen in those specific drugs?
So the cis bond actually is an amide bond between the carbonyl and the nitrogen. Okay. When I was pointing the structure, I was pointing the amide bonds because that's what protein looks like. It's a bunch of amino acids linked with the amide bonds. So most of these drugs have this feature. Specifically, most important is the site where it cuts, and this is between the phenylalanine and the proline. So phenylalanine and proline are the ones that have the bond that get cut, and then protein is separated, and so forth. Okay. So there are actually more than one site because protease has to cut more than one protein, but this is one of them. In this case, it mimics the phenylalanine proline site, and the drug was designed to mimic that such that the protease get confused and bind to it.
Okay.
Makes sense? Sure. Any other questions? Okay. Yet another example is ritonavir. The same thing, it's a protease inhibitor, acts on HIV, it's also highly active antiretroviral therapy, also acts on hepatitis C. Okay, what's unique about the ritonavir is it actually inhibits the CYP3A4. So that's, is that a good or bad thing? It's a bad thing, right? Because drugs, CYP3A4, what is unique about this enzyme? Most drugs are metabolized. Thank you so much. 50% of drugs in market are metabolized by this drug. So if you combine this antiviral with any other drug, there's a 50% chance that it's going to inhibit its metabolism. Okay? And so you've learned about how you manage drug-drug interactions. I'm not going to visit this again, but just something to keep in mind. Okay? On the other hand, that's been taken to our advantage in a different way. How is that? There is another drug, okay, called tipranavir. Okay? This is also a protease enzyme inhibitor. This drug is metabolized by this enzyme. Okay? So if you combine those two drugs, guess what? First, you're getting increase in the efficiency because you're combining two antivirals. Second, this one here is going to inhibit the metabolism of this one, and so you'll have more of this drug in the body. So you're combining, you're getting two advantages, you're getting two effects. The amplification of the antiviral effect, as well as you have more of the drug, this here in the body. It's not being eliminated, so you'll have more of the drug, higher concentration of this second drug. Make sense? Eric? And I'll get to you.
Yeah, like, could this be the reason why there's like the combination, I noticed this on TV. There's like a lot of like antivirals that are like this combination with this being this brand name. Is that why they're a combination?
Okay, so the question was, is that why we always combine antivirals? So actually, that's not the main reason, but that's a secondary reason. The main reason we combine antivirals is because viruses mutate very often. There's a good chance to get mutated viruses, unlike the bacteria. So to fight this mutation, we combine 2 or 3 antivirals together. Make sense? So that's the main reason. Of course, in this case, the second reason is this one is inhibited by, its metabolism is inhibited by this one, and so you would get a second, a second important factor, a second, important reason to combine them.
Okay.
Make sense?
Yep.
So the main reason for combining, and you would see that a lot, guys. Anytime we see, especially with HIV, they always use combination, 2 or 3 antivirals. I mean, like you can tell from the list of drugs that you're filling that this must have be like an HIV because they're combining 2, 3, 4 antivirals together just to prevent the mutation. Okay. In fact, Paxlovid, it has 2 drugs together, right? One of them is a protease inhibitor. Guess what's the function of the other one?
So maybe inhibiting with the
It's an CYP3A4 inhibitor. It has no antiviral activity. So we're combining 2 drugs. One of them has the antiviral activity. The other one has no antiviral activity, but it's just there to prevent the metabolism of the first one, such that the first one is staying longer in the body. Does that help? Does that make sense?
Yeah.
So that's applied in the Paxlovid. It's also applied here with this drug as well. I mean, not just by combining it with this one. Another way is by combining this drug by something called cobicistat, which is an inhibitor to the 3A4. This has no antiviral activity. Okay, it's only there to extend the effect of this drug. Everybody follows that? So in short, we have antivirals. They are combined to enhance their effect, but they could also be combined with other drugs that has no antiviral activity, but they're only there to prevent the metabolism of the antiviral drug. Okay, and that's very common. A common example is Paxlovid. Check the 2 ingredients for the Paxlovid and then you'll find out, okay? Which is one of the reasons why Paxlovid, if whenever a patient is taking Paxlovid, if they are taking other medications, you would see serious drug-drug interactions, and then you have to counsel the patient. You either have to stop the other drug or cut their amounts or adjust the dose or do something about it, okay? Okay, you had a question, you have a question here.
So earlier you said we could combine ritonavir and tenofovir.
Yes.
Do you recommend that when we combine drugs, they should be of different classes because that would work better for synergism?
Excellent point. So the question was, when we combine drugs, we should have drugs of different classes. That's true. In this case, I believe the combination is not just for the inhibition, but there might be binding at a different site of the protease. So remember, protease is a long protein, so we're chopping different parts of the protease. So by inhibiting one site, you're actually targeting one site, and then by inhibiting another site, you're actually amplifying the effect. Okay?
Since it's blocking CYP3A4, you have higher concentrations of ritonavir. Wouldn't that lead to even more adverse effects?
Excellent. So by inhibiting the antiviral, by combining these 2, one of them concentration is going to increase. Shouldn't that increase the side effect? Well, guess what? When you combine them, you would not give the same dose of each one of them. You would reduce the dose. So you would get the same effect, but you would not get the side effect. Okay? So instead of giving naturally you would give, let's say, 1,000 milligram. By combining these two, you would give 500 of each. So you're reducing the dose. You're getting the same amplified effect of antiviral, but you actually have less side effects because this concentration is going to reach to the other organs in less concentrations. And so you're not getting the side effect with these small concentrations. Make sense? In fact, I sometimes use that when I take Advil or Tylenol, and instead of taking the maximum dose of each, I could take 50% of each one of them. This way you have less side effect, but then you get the maximum effect from combining them. Right? They used to do that with kids, actually. I'm not sure if you noticed, there was a time where they said every 4 hours you give Tylenol, then you switch to Advil and back and forth. However, apparently people got confused, and so they were giving the same drug every 4 hours, and then they got into toxicity. So they removed this protocol. So as pharmacists, if you understand the protocol, you could apply it. But people who I guess don't pay close attention, they might, or sometimes they might combine Advil with Motrin. They think, even though they are both the same, but people who are not pharmacists, they might not realize it's the same ingredient. But the concept, pay attention to the concept. Tylenol, Advil, they work on they have two different side effects. They both help with fever. If you alternate every 4 hours, you get the maximum effect with less side effect. And that was the protocol. However, people got confused and they got into repeating the same drug twice, and there was problems, so they eliminated this protocol because of the confusion. But you could use it as pharmacist, okay? Experiment on your kids, I guess. Say that again.
For long-term use and stuff, but we don't often see topranavir with ritonavir actually used together. Is that because of the liver toxicity?
You don't see the tipranavir and the ritonavir combined together? You don't? Oh. I'm not sure if the clinical studies have shown something different, but in principle, you would see the combinations of these drugs like that. Okay. So I'm sure with clinical, you would see different protocols, or maybe one protocol recommending different way of combining drugs, but I care more about the concepts, okay? Any other questions? All right. Okay, so we have covered all these. Next we have fusion inhibitors and integrase inhibitors, and 304. Do you need to have a break and come back? Break? Okay. So if you need a break, raise your hand. If you want to continue, raise your hand. Okay, so I think more people need a break. Sorry. All right, so let's have 10 minutes break and come back, okay? There's not much though. There's only, let's see, 4 more pages. So anyway, you want to continue? Yeah? Wait. Okay, let's have 5 minutes break. 5 minutes, not 10 minutes, okay?
Dr. Kashan, just one question, right? You mentioned something about the protocol that you mentioned. And is it okay if you can go and repeat that again? I just wanted to make sure to get that understanding.
Sure. So the idea was, there was one time when they thought for kids, instead of when they have really high fever, instead of having to stick with one drug, let's say Motrin, and take it constantly, because if you take Motrin, you have to do it every 8 hours. But then before the 8 hours, 2 hours before that, you get the fever. So you cannot give Motrin earlier. So they say, well, let's switch to Tylenol, and then that will buy some time before the next time you get Motrin. And so they come up with protocol that says, okay, you get Motrin, and then after 4 hours Tylenol, after 4 hours Motrin. Okay. So this would separate the Motrin from the next dose by how many? 8 hours.
Yeah.
It would also separate Tylenol from the next dose by 8 hours. So you achieve the goal of controlling the fever, but without getting the side effects from each.
Oh, so it's like you
Was-
split
was
them for like
brilliant.
8 hours? You separate them between 8 hours.
No, you separate it. So you take Motrin, 4 hours take Jamo, 4 hours take Motrin, 4 hours
Oh.
take Tylenol.
Oh, I see.
So in theory, you're separating Motrin by 8 hours, and you're also separating Tylenol by 8 hours, but they're constantly giving the anti-fever. It was a brilliant idea. However, people got confused. They were sometimes giving Motrin and then go Motrin again, or they forgot what I gave them last time.
Right.
So there were so many mistakes. So they
Ah, I
eliminated the protocol. They thought, even though it works, but not for the I guess average size. So they stopped.
Oh, I see. This is only for like fevers and doses of medications, right? It's not like for anything else.
So
Clear?
this is primarily because like kids when they get the infection and fever, and you can't really control it constantly because so they can only this thing. I mean the concept itself implies like you get less amount of control, maybe less than less frequently. So you get the effect and then less side effect. That was the use.
Oh, I
Use this
see.
as an analog. When you combine two drugs, you actually get you affect the dose from each one of them, so you get less side effect.
I see. Okay. Yep, it does. Thank you so much.
Is everyone back? Are you missing anyone? I mean, do you see anybody around you who's missing? No? Okay. Okay, so next we'll talk about the fusion inhibitors and integrase inhibitors. All right, so just a reminder, fusion is when the virus attach itself to the surface, okay? So apparently we have some receptors from both sides that interact, and then we're gonna interfere with that, okay? So the first example on the fusion inhibitor is something called enfuviratide. I mean, as you can tell from the name, it acts as an fusion inhibitor, okay? And it actually acts on the CD4, interferes with the CD4 and the GP41. So GP41 from the human, CD4 from the viral, they interact with each other. So this one binds with the GP41, sorry, 41, not 4, okay? Now this drug is actually a protein. It's a polymer, oligopeptide. It has 36 amino acids, okay? So, I mean, just by looking at the structure and the fact that it's a protein, can that be taken orally? No, it has to be IV, okay? It has to be taken IV. It's like just like insulin or any of the protein drugs. So this one here interferes. So if there is a receptor from the human body and then another one from the virus, this one interferes in the middle and then it blocks the interaction and prevents the fusion, okay? As simple as that. Clinical applications for HIV-1, HIV-2, side effects. There is some injection site reactions such as itching, swelling, redness, okay? There is some also things like nerve pain, numbness, burning, prickling feeling of the skin that could last up to 6 months. Okay, so it's mostly has to do with the site of the injection, okay? So that's IV. The next one, which could be taken orally, is the Maraviroc, and it interferes with the CCR5, the human receptor CCR5. Again, the same concept. It interferes with the receptor that interacts with the enzyme, and then it blocks the fusion. This drug is substrate for the CYP3A4. So as you can imagine, inhibitions to this drug could result into more amount of this accumulation of this drug. It's also a substrate for the PGP pump. You remember, guys, the PGP pump? What's the function of the PGP pump? It efflux kicks out the drugs out of the system in 4 different positions: in the intestine, kidney, and liver, excretion sites, and also in the blood-brain barrier. It protects the blood-brain barrier. It protects the brain, so it's in the blood-brain barrier, kicks the drug outside. So by blocking this, the drug could accumulate. Okay? And so there might be some interactions with some drugs that interfere with that. You don't have to memorize the examples, of course, just the concepts. Clinical application for the HIV type 1, side effects: headache, fatigue, diarrhea, nausea, upper respiratory tract. Other most common adverse reaction in studies are upper respiratory tract infection, cough, pyrexia, rash, dizziness. Any questions about this class? So we talked about 2 examples. One is actually a protein, so it has to be injected. The other one is small molecule, could be taken orally. Okay? All right. Just make sure you know the mechanism. So this one is acting on the CCR5. This one is acting on the GP41. Okay. All right. The next one, which is the last class, are the integrase inhibitors. So as we said, at some point, the DNA piece has to be inserted into the human DNA, and the enzyme that does that is the integrase. So we could inhibit this enzyme. Okay. So again, this is the DNA of the viral being inserted into the DNA of the human. So we have integrase inhibitors refer to INIs. The example we have is something called raltegravir. Okay, so we talked about its mechanism of action. It's used along with other medications to treat HIV infection. So, the combination of antivirals, and these are the side effects. Okay, and that's the only example I have for the integrase inhibitors. And this table is just for your information, like a summary of the drug and its mechanism of action and the viruses or diseases that are affected by it. Any questions about this category or anything else? Any questions? Yes, Eric.
You mentioned something about like this being an integrase inhibitor. So what does it necessarily do in terms of that little diagram that you showed?
Oh, so there is an enzyme called integrase that integrates the DNA into the viral DNA into the human DNA. So it blocks the enzyme, it inhibits the enzyme. It inhibits the integrase, okay?
Okay, so it's like an enzyme that's just able to block that.
Yes, so it's integrase inhibitor. Any other questions? So far so good. Any questions about the exam? So hopefully it's clear what you need to study for. If you're unsure, shoot me an email, okay? I know when you guys have exams, I pay close attention to my emails. So even it's just a simple email, don't waste time on things that you probably shouldn't be studying. Like if you're not sure, should I memorize that? Should I study this table? Before you do that, just shoot me an email. If you're lucky, I'll be online and reply immediately. Otherwise, you will get some answer before you spend time. Yes?
About the exam, for the side effects, should we know any of them that are not bolded?
The ones that are not bolded,
Yes.
I want to be careful. I'm afraid to say you shouldn't, but I mean, only the ones that we spend time and like emphasizing. But if I go like on the list, like, just like now, for example, we said, these are the side effects, then this and that, then it's nothing important. But if I stop at something and say this causes this side effect, which is important, or like, then you would need to catch it, okay? If you're not sure, again, just shoot me an email, okay? Yes.
The medchem for today was the selectivity of the drugs.
Well, it's like a mix of like both medchem and pharmacology. So then, like because I'm teaching both, it won't be like separation. But your questions in the medchem, but the structures, not memorizing them. But for example, I do want you to know that this is a guanosine analog versus cytosine analog. You can tell the guanosine has 2 rings, cytosine has 1. I want you to know that this is nucleoside analog. This is non-nucleoside analog. You can tell from the structure that it has no, guanosine or cytosine, right? What else? So, I mean, some information you could derive from structure. The ones we've seen where it has, this huge molecule. I mean, there's no way this could be taken orally, right? So this must be taken by IV, right? So relate the structure to its activity, I guess. Other questions? Any other questions? Last call for questions. Okay, just shoot me an email if you're not sure, okay? All right, have a good afternoon, guys. See you next week or so. Thank you.